You're watching Tumor TV. Knowledge is power.
So tumor DNA testing, as we described that previously, basically what happens is we take a sample of your biopsy, or we can isolate tumor DNA from the blood in a new process called liquid biopsy. What we do is we isolate these tumor cells, and then we extract the DNA from them. And then we look at about 500 or 600 different genes that are involved in cancer that we have targeted therapies for. And we find out what mutations are causing your cancer. And then we can come up with a combination of drugs that target those specific mutations. So chemotherapy works a little differently. Standard chemotherapy, what it does is when it gets injected into you, or when you take it in a pill form, what it does is it kills any cell that happens to be rapidly reproducing. So most chemotherapy drugs interfere with the cell cycle. The cell cycle is just a reproduction. It's like having babies. It's when a cell becomes pregnant and with its daughter cell. They don't become pregnant. What happens is they split in half and divide and share their genetic information. So most chemotherapy drugs will interfere with that. However, the problem with standard chemotherapy is it's only effective when it's in your body, and it's only active in your body for typically a few hours. Some chemotherapy drugs will last, 10, 12, 24 hours, but they only affect the cells that are rapidly reproducing at that time. So if you have a tumor that just doesn't happen to be growing at that particular time, then chemotherapy is not going to affect it. And it's also not going to affect the stem cells that cause cancers too. So knowing what DNA mutations you have is really important, because then we can use targeted therapies and not have to rely only on chemotherapy.
Thank you for watching Tumor TV. Subscribe below and stay informed.